Okay, let's get started. It's really my pleasure to introduce my colleague and good friend, Darren Roblyer. Darren is a professor of biomedical engineering at Boston University. He's really done a lot of work in using label-free optical techniques for disease diagnosis and monitoring therapies. He completed his undergraduate degree at Johns Hopkins and then went on to Rice University to earn his PhD under the guidance of Rebecca Richards-Cordham there. And then I got to know Darren when he started his postdoc here with Bruce Trombler. And so there was, I guess, about 3 years, I think, overlapped. And then he went on to BU to establish his research group. Over the years, he's really pushed both the instrument development as well as clinical applications of a number of modalities in biophotonics, ranging from spatial frequency domain imaging to frequency domain near infrared reflectance and spectroscopy. And then more recently, moving into shortwave infrared imaging and speckle contrast optical spectroscopy. And he's really working on instrument development, wearables, a number of applications ranging from breast cancer, kidney disease, rheumatoid or autoimmune disease, and cardiovascular disease. So this is the first of 2 lectures that will be we'll get the pleasure of listening to, all focused around the use of multiply scattered light, kind of in the transport to diffuse regime, to give us insight to tissue structure and physiology. So let's welcome Professor Roblyer.
Thanks, everyone. It's really nice to be here and to see you. And big thanks to Vasa and Bernard for organizing this 2-week short course, which isn't so short. It's really a ton of effort to organize this and make it work. So make sure you thank them before you leave. And it's a pleasure here to kind of help finish up the end of the short course. So as Vasa mentioned, I'll be lecturing today and then tomorrow morning again on diffuse optical techniques, kind of focusing on the 4 techniques that are most commonly used in the diffuse optics realm. And so I'm going to be here for a little while, so you might as well get to know me, which means feel free to interject, ask questions. Let's have a discussion. I'll ask you a few questions during my talk. And I thought I'd start by telling you just a little bit more about myself. So Vasa mentioned, I went to Hopkins for undergrad and then Rice as a PhD student. When I did my PhD down here, it was with Rebecca Richards-Cordham. I focused on cervical cancer, although my thesis ended up being in oral cancer using autofluorescence techniques to identify margins, surgical margins, so you can remove an oral lesion while preserving as much tissue as possible. Then I came here and worked with Bruce on frequency domain diffuse optics, which we'll talk about today and tomorrow, as focused on the application of breast cancer and chemotherapy monitoring. And then I started a faculty position. When was that? 2009. I've been at BU for 13 years. And I think you'll hear even more from me because I'm on the academic career panel afterwards. So we'll talk more about that and if you have any questions about that. And BU's been great. This is Boston. This is the Charles River. Boston University is a campus that's along the Charles River. It's very long and narrow. My building's here. You can see Fenway, the baseball stadium over here. This is the Prudential Center, and this is Boston on a particularly nice day. This is my lab. The picture's a little bit old because I do have a student here, Anna, who's not represented in the picture. So we need to retake it and include her. But we focus all on diffuse optics, both in the near infrared and the shortwave infrared, as well as speckle-based imaging. We have funding from a number of sources. I'm also the graduate chair for our department, so that means I oversee the PhD program for our 200 plus PhD students. And I was the grad admissions chair before that. I'm also the editor-in-chief of a new journal from SPIE called Biophotonics Discovery. So this is a journal that's focused on basic science and clinical translation, all associated with biophotonics. And you might want to think of it, you're probably familiar with the Journal of Biomedical Optics, another SPIE journal, and maybe the Journal of Neurophotonics. But JBO, I've been thinking of it more like methods, instrument, and then biophotonics discovery is a little bit more about what you're going to do with those emerging technologies. What can you discover in the basic science side, and then what happens in your first in clinic or with clinical studies. And so you can see the 2025 call for papers. We have 6 of those open right now. This topic, maternal reproductive and neonatal health, that's going to be the focus of the Sunday plenary at Photonics West. It's also a new conference that's going to be part of Photonics West on that topic. I also wanted to mention, because I know many of you are PhD students and some postdocs, we have an early career editorial board. So this is for postdocs instructors, and we use them as a reviewer pool, but we also have our eye on them for future associate editors. So as you become a postdoc or an instructor, we can also consider equivalent positions in industry. Feel free to contact me and let me know if you're interested in being an early career editor. We want the journal to also be a mechanism for career development and have exposure to publishing. And I know Gwen came last week and talked to you guys, right? Gave you 2 lectures on publishing. And I work very closely with Gwen on starting the journal, which is only about 1.5 years old. Some disclosures before we start. I do some consulting for Aura Health. Do you guys know what Aura is? You know what the Aura ring is? I have one on my finger. It's a wearable that goes on your finger. I think it's a very cool technology. I'm also a co-founder of a new company called Rivis Optics. Okay, but to the topic of the lecture today. So the course is about biophotonics. My lab focuses on translational biophotonics, so measurements in humans. So let's look at what the current tech is. And so what I mean by that is, let's start with the technology that it's actually made it into the standard of care. So not just in the academic realm, but actually changing how patients are treated and diagnosed. We have things like endoscopy, laparoscopy, difficult to say, where you can look at the inside lumen of organs like the esophagus or the colon. Surgical imaging, where you'll have magnification to improve the accuracy of surgery. Oximetry, which is more in the diffuse optics realm, where you can get tissue oxygen saturation. Dermoscopy for skin lesions, and things like OCT, optical coherence tomography, for looking at the eye. This is actually a huge field. The market size, 128 billion, estimated to be the single largest technology sector in medicine today. And these technologies are amazing, but you might notice that they all seem kind of like maybe yesterday's technologies, right? You don't really see contrast agents. You don't see photoacoustics. We just had a lecture about earlier today. OCT is maybe the most cutting edge, but the application that's really taken fire is in the eye, but OCT has many other applications, few other diffuse optical techniques. So all that to say, it's just a big filter to get from academia or research lab and industry all the way to clinical translation that can take decades. So there's so many new developments that you'll all be working on that we hope to get into change clinical care. It also means there's tons and tons of opportunities. And in the diffuse optics realm, we focus more on oximetry. The most famous example is pulse oximetry, where we get peripheral arterial oxygen saturation, but we're going to talk about many more. And to differentiate diffuse optics from other types of imaging like microscopy, you know, if we take a look at this picture where we have this tissue-like media and we shine a laser into it, this first part of the diagram and the first few hundred microns, that's really the realm of microscopy, where photons haven't had a chance yet to scatter multiple times. And you're really trying to reject multiply scattered photons to get high resolution, high spatial resolution images. Diffuse optics are when the photons have traveled long enough so that they start to propagate almost isotropically. So here we have lower spatial resolution, but the upside is we have a lot of functional molecular information that we can use as contrast. So here's an example of confocal microscopy with or without the pinhole that rejects the multiply scattered light. That gives us very high resolution but shallow imaging depth images. In diffuse optics, we can get light all the way through tissues and organs, millimeters to centimeters deep, but lower spatial resolution typically. And in terms of translational and label-free diffuse optics, you know, we have some version of an optical source and an optical detector measuring some part of the body. We have no contrast agents here. This reduces regulatory burden, both in terms of, say, the FDA and trying to get something FDA cleared, but also in academia. The IRB protocols you have to write tend to be less laborious than those that would require an exogenous agent, for example. Cost-effective and non-harmful, and we have key contrast. So a few examples of technologies that fit into the realm of diffuse optics that I'll talk about in the next 2 days are FNIRS, functional near-infrared spectroscopy, where you're using near-infrared light to measure the spatial location of functional activation in the brain. Diffuse optical spectroscopy, which is a bit of a catch-all. It can mean continuous wave and frequency domain and time domain techniques where we extract optical properties and then chromophore concentrations, oxy, water, and lipid. Shown here, this is a 3D rendering of the breast where you're actually looking at real clinical data that shows the contrast of a tumor. Spatial frequency domain imaging, where we have a non-contact version of diffuse optics, where we can map optical properties, in this case over the hand. What you're looking at is a healthy volunteer in this column and a patient with a disease called scleroderma, where collagen builds up in the skin and actually causes a reduction in optical scattering. That's what you're seeing here. This is why this image is darker than the associated healthy tissue. Tissue oximetry. So there's commercial devices that you can buy, like this one from Bioptics, where you can put it on a skin flap. This is on the breast, and it tells you how oxygenated or perfused that skin flap is that's been surgically implanted. Pulse oximetry, which I mentioned, and then wearables and remote patient monitoring. These devices can currently measure things like your heart rate, SpO2, breathing rate, and in the future may be able to measure other things like your hydration level, body composition, meaning how much lipid and water you have in your body, as well as blood pressure. And there's the Oura ring as an example of that. So diffuse optical contrast range from functional. We can get the function of the tissue from oxy and deoxyhemoglobin. We can get structural information. Scattering tells us about the microarchitecture of the tissue. Collagen and elastin are chromophores that we can extract that also tell us about the structural components of tissue. And molecular information, water and lipids, same contrast that you'd get from MRI. That information tells us about the makeup of the tissue, gives us molecular contrast. The learning objectives for today and for tomorrow are for you to gain an intuitive and quantitative understanding of how diffuse optical technologies work in practice. We'll leave more of the theory to Vasan, and you've already had some lectures from him and others on theory related to diffuse optics. And I also want to cover what are their capabilities and limitations. So we'll keep an eye on these learning objectives as we go through. Topics we're going to cover today. I know you've already talked about optical properties. We're going to review some basic optical property facts briefly, then talk about chromophores, some versions of Beer's law and the Beer-Lambert law, and then focus on two different types of diffuse optical techniques. Spatially resolved measurements, where we'll talk about the fundamental principles. We'll talk about inverse models, which you need to extract optical properties and chromophores from tissue. Different versions like the radiative transport equation, the standard diffusion approximation, and Monte Carlo-based inverse models. The near versus the SWIR, which I'll define in a moment. And then we'll switch to a different but related topic, spatial frequency domain imaging. Principles, and if we have time, we'll talk about wavelength selection. Okay, here we go. Oh, I should mention other diffuse optical techniques, clearly defined as diffuse optical techniques that I won't cover today or tomorrow, include DCS, SCOS, and LSI. And this is largely because I believe you've already had lectures on these technologies related to blood flow and less related to extraction of optical properties.
You know, less than they did it yesterday.
Bernard, you gave a lecture on speckle, right? Yeah, I'm sure it was excellent. Okay, optical absorption and scattering. Absorption quantified through mu-A, the absorption coefficient, which is in units of inverse distance. In my lab, we quantify in terms of inverse millimeters, but others will quantify in units of inverse centimeters. Just be aware of that difference. Absorption in tissue is caused by chromophores and molecular species that absorb. Here's a list of them that absorb in the visible, the near-infrared, and the shortwave infrared. Scattering, quantified by the scattering coefficient, also in units of inverse distance, caused by refractive index changes, things like the difference in refractive index between the cell membrane and the cell nuclei. Tells you about cellular density, morphology, microarchitecture. So mu-A defined as the probability of absorption per unit path length. Likewise for scattering, right? So if your mu-A is 0.1, that means if a photon travels a millimeter in tissue, there's a 10% chance on average that it will undergo an absorption event. So similar with scattering, total attenuation, mu sub t is mu-A plus mu-S. A few definitions, wavelength bands. These wavelength bands don't have consistent definitions in the literature. For the lecture today, we'll call the visible from 400 to 650, the near infrared 650 to 1,000, and the shortwave infrared 1,000 to 2,000 nanometers. In the literature, you'll see plenty of sources call this 1,000 to 2,000 nanometer still the near infrared. And then within these bands, some will refer to them as optical windows. For the near infrared, for example, we call that the first optical window. And then different portions of the sphere we call the second or the 2A and 2B optical windows. And you can see some of those definitions here. But the optical properties of tissues, biological tissues, are different in these different wavelength bands. And the goal for the diffuse optical techniques I'll talk about today is to, in many ways, extract chromophores. Chromophores meaning the things that absorb light in tissue, and use those as biomarkers to make clinical decisions. So let's take a look at this figure. Here we have wavelength versus mu A. And this is for the example of a healthy breast tissue. And you're looking at the absorption spectra of oxy and deoxyhemoglobin, lipids, and water. These are not the only chromophores that absorb through this broad wavelength range. Melanin, collagen, elastin, beta carotene under some circumstances, but they are four that occupy the majority of absorption through many wavelengths. You can see their concentrations here. This is just from literature. Like these are typical values for healthy breast tissue. 10.42 micromolars of oxy, 5.27 deoxy, water represented as percentages of pure water and lipid. They have molar concentrations associated with them, but we often represent them as percents of pure water or lipid. And you can see that these absorption shapes, they have specific shapes to them. They have peaks and valleys, and they're lower and higher in some areas than others. And tomorrow I'll say a little bit more about what actually causes those peaks and valleys. But what we typically want to measure is the total tissue absorption. Yeah.
The percentages you show, are they volume fractions or is it like something else?
Yeah, volume fraction. So in your measurement volume, you have a molar concentration, moles per liter of these different chromophores.
Okay. Okay, thank you.
Yeah.
Other questions?
So we want to get at, for example, we'd love to measure this tissue and be able to extract, to say, well, this tissue has this much oxy, this much deoxy, water, and lipid. The way we do that typically is by measuring the total tissue mu A at multiple wavelengths, then disentangling how different chromophores could add up to contribute to that total mu A, right? So that's what we measure, the dashed black line. And it'd be rare for us to take a tissue measurement through this entire wavelength band, right? Often you focus on the visible, the near infrared, or the shortwave infrared. But you need to measure at least a few wavelengths in order to extract the chromophore concentrations. And here's the first law I'll show you, Beer's law. So what we measure is mu A, units of inverse distance. We want C, the concentration of a given chromophore. Let's call it oxyhemoglobin for a moment. And then this relationship between mu A and the concentration is dictated by the extinction coefficient, which we can get from literature. So concentration, moles per liter, extinction coefficient, liters per mole per millimeter. And that has the units work out so that mu A is in units of inverse millimeter in this case. Is anybody wondering what this 2.303 is? I'll speak a little bit more about it tomorrow, but this is just a conversion. Extinction values you get in literature, and I'll show you a whole bunch of sources for extinction values tomorrow. But often extinction values in literature are given in log base 10, but this Beer's law and mu A is defined in log base E. So the 2.303 is just the natural log of 10. It's a conversion between the extinction coefficient in log base 10 to log base E. Okay, you can see the simple linear equation. Now we typically have multiple chromophores, so this can be in vectorized form. So mu A, let's say we measure mu A at multiple wavelengths. We have extinction coefficients here. This column is for deoxyhemoglobin. You have different extinction values for different wavelengths per chromophore, oxy, lipid, and water. And then we solve for these concentrations, only one concentration for the tissue volume, right? That's not wavelength dependent. And these chromophore concentrations, and sometimes mu A and mu S prime, are what we use as biomarkers to make clinical decisions. It's what we're using to make diagnoses or prognoses for a given subject. So we want to extract those chromophores. I say sometimes mu A and mu S prime because sometimes scattering is the metric you're actually using. You say if you're above the scattering metric, maybe you have disease X. If you're below, you don't. Now, mu A and mu S prime, again, probability of scattering per unit distance. I want to talk a little bit more about scattering. Scattering in tissue tends to be very forwards directed. So have you talked yet about G in the course? G. Okay, so we'll go over it briefly. But G is called the anisotropy factor. And in a single number, it tells you how forward scattering the light is or directional the light is. Right? So it's just the average cosine of the scattering angle. If you have a photon coming from the left and it scatters directly ahead at what we call 0 degrees, the cosine of 0 degrees is 1. So in that case, if that was the most likely angle, your G value would be 1. And if you diverge just a little bit from 0, your G value would be high. So the range of possible G values is between 0 and 1. And many tissues are between 0.75 and 0.95. And again, this is a value that's averaged over many scattering events. So one way to think about this is it takes several scattering events to significantly change the direction of a photon as it travels through the tissue. And we define the reduced scattering coefficient as 1 minus G times mu S. So if G is typically around 0.9, 1 minus 0.9 is 0.1. So your mu S prime is about a tenth of mu S. It's one reason it's called the reduced scattering coefficient. And this is a figure from a very famous and useful review paper on tissue optical properties from Steve Jacques from 2013. And you're looking at this anisotropy factor G for many different publications and tissues. And you can see that it hovers, not in all cases, but many cases at very high values, like above 0.8 to 0.9. So oftentimes we'll extract mu S prime, which encodes information also about G in it. And then I think it's very useful for you to have in your back pocket some optical properties of tissue that you can use to make sense, make sure you're in the right order of magnitude. So for my students, I tell them, think in the near infrared, many tissues have an absorption coefficient of around 0.01 inverse millimeters and around 1 inverse millimeters for reduced scattering. Right? So I want you to remember those numbers. This is useful because you can already see that scattering is about 10 times or 100 times stronger than absorption. Right? So if photons traveled over a millimeter, only 1% chance of absorption and 100% chance of scattering. And then let's look at some specific tissues and how optical properties change in different wavelength bands. So these are optical properties from literature. Sources are shown here, but what you need to know is just brain, breast, and skin. 3 different optical tissues to show you some variety. And in the figures, you're looking at the visible in blue, near infrared in white, and orange in the shortwave infrared. This figure is showing absorption on log scale. And what you can see visually is that there's lower absorption in the near infrared. It's one of the reasons we call that the optical window. Absorption is quite low, so your photons can make it a little bit deeper into tissue because they're not being absorbed as quickly. And the near infrared mu A tends to be lower than both the visible and the shortwave infrared for many tissues. This is reduced scattering coefficient. It follows this relationship, an empirical relationship that can be fit the power law in many cases, especially in the near infrared and shortwave infrared. So the shortwave infrared has the lowest scattering. And then if we look at the ratio of mu S prime over mu A, that ratio is highest in the near infrared. This becomes important when we talk about the diffusion approximation and diffusion theory, where we want to apply it in media where mu S prime is sufficiently large compared to mu A. So for example, this dashed line is a value of 10. Use the diffusion approximation for tissues when that value is at least 10. We often like a value of 100. So these differences have a profound effect on how light travels through tissue. Diving a little bit deeper into this, I showed you Beer's law. I'd now like to introduce the Beer-Lambert law. So let's pretend we have some media that only has absorption in it. Remember, our end goal is to get at these chromophores. To do that, we need to quantify absorption. And in this example, we send in light into this media. We measure light that comes out the other side. This media has some thickness to it. We'll call it L. The Beer-Lambert law gives us this relationship. It shows us that it's an exponential relationship. We lose light intensity as a function of natural log of E, the factor of mu A times L. L is the path length, but in this case, there's no scattering, so the path length is just the thickness of this media. So you can actually extract optical properties of materials this way, as long as there's not too much scattering. I'll show you a little bit more about that in the next slide. This relationship actually holds for scattering as well. Suppose you don't have any absorption, but you do have scattering, and you have this baffle here, so photons only make it through this area here where you don't have a block. As scattering increases, along with the path length, this relationship holds. And if you have a media that has both absorption and scattering, you can use mu sub t, the total attenuation, mu A plus mu S. So this is a figure a postdoc of mine made a few years ago. He took a cuvette, put water in it with a little absorbing dye, sent a laser beam through it. And then as you go from left to right, he added more and more titanium dioxide. This is a nanoparticle that scatters a lot like tissue scatters. And in the left-hand side, you really have very little scattering. Laser beam cuvette. You could quantify mu A just by knowing the thickness of that cuvette and measuring the light intensity in and out. But tissue is much more like this. An essential challenge is that we can't really know the path length very easily. The path length of these photons is going to be circuitous as it makes it from one side to the other because of scattering. And there's a challenge in disentangling absorption from scattering, which is why we have a number of different modalities in diffuse optics that allows us to do that. And so a comparison of the diffuse optical techniques, this is 3 out of the 4, introduce the fourth later. But the first is continuous wave. So continuous wave, and we'll combine that with what we call spatially resolved measurements. We send light of a constant intensity into tissue, measure light coming out of the tissue. This could be in reflectance or transmission geometry. And that attenuation of the light is going to be due to a combination of absorption and scattering. That means this technique has some limitations. We can measure the delta, the change in absorption. If we assume scattering doesn't change, or change in hemoglobin or oxygen saturation levels. We can extract mu A and mu S prime under certain circumstances with specific constraints. And I'll show you some examples of that. Other techniques where we modulate the light source, what we call the time frequency domain or the time domain, give us sufficient information to separate the effects of absorption from scattering. If we do that at a number of wavelengths, we can get chromophore concentrations of deoxy, water, and lipid, and others. So let's start with continuous wave spatially resolved measurements. Okay, the continuous wave is referring to the fact that we're not modulating the light source. Spatially resolved is referring to the fact that we have some source and then typically multiple detectors at different source detector separations or rows, that symbol rho, away from the source. And let's look at this relationship. On the y-axis is log scale, diffuse reflectance, which is a value that's scaled between 0 and 1. I'll talk more about what diffuse reflectance is later, but for now, just think of it as related to the measurement intensity at the photodiode. And then on the x-axis is distance from the illumination point. So you can see the illumination falls off, or the detected intensity falls off as a function of distance from the illumination point. And the 4 lines are showing you 4 different optical property combinations. Low absorption, low scattering, high absorption, low scattering, low absorption, high scattering, and high absorption, high scattering. We're going to use these 4 scenarios to see how different types of measurements help us disentangle absorption from scattering. And so for spatially resolved, it's actually a little bit tricky. Let's take a look first at the high absorption versus low absorption cases. You see the arrows pointing to those lines. You can see that there's some overlap, right? This low absorption line falls off faster than this high absorption line. If you take a look at scattering, there's a little bit more separation, but this low scattering and high scattering line aren't on top of each other, but they're close. This brings us to some of the advantages and disadvantages of these types of measurements. They're relatively simple and low cost, miniaturizable, because your light source could be an LED, for example. Your detector could be simple photodiodes that are in small packages that you can put into a wearable. They can be battery operated and fast. But disadvantages include the fact that absolute quantification of absorption and scattering is tricky and not always possible. It requires some assumptions and constraints. Look at these two scenarios. So you have high absorption and low scattering, and low absorption and high scattering. Those lines are almost on top of each other, difficult to disentangle. I also want to focus on measurements really close to the source. Do you see here that the 4 lines collapse basically into 2 lines? This bottom one is the low scattering case that includes both a high and low absorption. And then this one is the high scattering case. So why does absorption seem to have no effect on measurements so close to the source? Who could give me an intuitive explanation of why that's the case? Who's got an idea? So really close to 0, if you take a measurement really close to the source, absorption seems to have no effect on these lines. Do you see how we don't see 4 separate lines, we only see 2? The only thing that's changing the measurement is the scattering, right? Differences in absorption in the tissue or whatever media we're looking at aren't actually changing your measured value. Yeah.
Is it because the like the mu A is so small, like you're traveling such little length, it doesn't have, I guess, distance or time to get it forward?
That's exactly right. So what are the optical properties I just suggested that you try to remember? What's a typical 0.01 for absorption, right? And scattering, reduced scattering? 1, right. So think about those metrics again over a distance of a millimeter. And look, this is 0 to 1 centimeter. So a millimeter is right kind of where we are. Now, of course, photons are going to take a circuitous path to get to the detector, but let's pretend that they travel a millimeter. With those optical properties, just traveling a millimeter, 0.01, 1% chance on average of undergoing an absorption event for that path length, and like 100% chance of undergoing scattering. So absorption has almost no effect on your measurement. And actually, people have made entire careers in the biophotonics community focusing on those really short source detector separation measurements to measure scattering in tissue without the effects of absorption changing their measurement. It's called elastic scattering spectroscopy. Irving Biggio at Boston University is really focused on those types of measurements. So that's one type of what we call path length gating, right? You know that if you can somehow measure photons that have undergone shorter or longer path lengths, you can be differentially sensitive to absorption versus scattering. But here at longer source detector separations, it's trickier for spatially resolved domain. Okay, and this was just a reminder of those optical properties I asked you to remember. So we say at very short source detector separations, we're scatter dominated, and at long source detector separations, we're scatter and absorption dominated. So let's say you build a device, a spatially resolved device or a continuous wave device, and you want to process your data that you're taking on tissue specimens or in vivo measurements. How do you do it? How do you get at optical properties? I'm going to go over 4 different potential ways to do it, starting with the diffusion equation, then next the modified Beer-Lambert law, then Monte Carlo-based inverse models, and then finally neural networks. So let's start with this diffusion equation. And in order to enter that, we want to first understand forward versus inverse models. So a forward model in this case, those terms have definitions in different fields. A forward model would be the case where we start with optical properties and a source detector separation, and we use a model like the diffusion equation to get out diffuse reflectance. That's our measurement. And turns out it's hard to solve the diffusion equation the other way around, where you input the diffuse reflectance and it spits out mu A, at least analytically difficult. So we typically wrap this up in a solver where we iterate, we guess some initial optical properties, we get a predicted diffuse reflectance, we compare to see if that predicted diffuse reflectance is close to our actual measured data. If it is, then we can say, okay, our optical property guess was about right. That's the answer. If not, we update the guess, hopefully in the correct direction towards the right answer. And this is how we get at an extraction of mu A and mu S prime. Now, Vossen showed you this slide, I believe, last week. Yes, I don't know if people remember it. Yes. I'm not going to go through the RTE, radiative transport equation, or also called the radiative transfer equation, also called the Boltzmann transport equation. There's many names, but it's a bookkeeping equation that tells you about if you care about radiance and photons that are traveling in a specific direction in a specific location, that time rate of change is due to streaming, loss of interaction from absorption, gain through scattering. A photon from a different direction can move into your unit direction that you're interested in, and gain from sources. So right, this I consider the mother equation, although you might say Maxwell's equations are really the mother equation. But in diffuse optics, we'd love to be able to use the radiative transport equation. It's difficult to solve. Some people try to do it, but we use the diffusion approximation where we make some assumptions. We expand the radiance into two terms that are the 0th and 1st Legendre polynomial, fluence and flux, which tells us that light propagates mostly isotropically with some directional flux. This is why it's an assumption, because under certain conditions, light will travel very directionally, especially close to the source, because G is so high, right? You're very forward scattering. You also use G value here, anisotropy, for scattering. But at the end of the day, you get down to this diffusion, also called the P1 diffusion approximation. P1 referring to the fact that you go up to the 1st Legendre polynomial. You can keep going the 2nd and 3rd, and you get closer to approximating the radiative transport equation. But you can solve this for different domains. So for example, you can solve this for the spatially resolved domain, and those solutions are in literature. And just a note here, so the terminology can be different depending on the source. We call the P1 diffusion approximation typically the standard diffusion approximation. I'll refer to it just as the diffusion equation in this lecture. And this looks nasty, but it's actually a closed form solution that relates diffuse reflectance as a function of source detector separation rho to these terms that are all just a function of the tissue optical properties and a little bit about the boundaries, refractive index of the media, and then the media above where you're trying to get the optical properties, right? So if you look here, everything you see, like Z naught is just 1 over mu S prime. Mu effective, here's mu effective. It's a function of mu A and mu S prime. So you can code this up in a few minutes in Python or MATLAB and start analyzing data. You have to wrap it up, and you can start extracting optical properties for a given solution to the diffusion approximation for given boundary conditions. This one's for semi-infinite geometry, where you're going off infinitely in X and Y and Z, but you have this boundary between your measurement device and, let's call it, the tissue. There are other more complex geometries and solutions for the time and frequency domain, and those are in literature, and we'll talk a little bit about them as the lecture goes on. Yeah.
My question is, does this equation work for smaller source detector distances, or is there like a limit?
Yep, I'll show you some figures associated with that, and I think Vossen also will in his lecture tomorrow. But the short answer is no. It doesn't work well for short source detector separations, like the really close ones I showed you before. You're not what's called in the diffusive regime.
Mm-hmm.
You're too close to the source, where light is not propagating close to isotropically.
Thank you.
Yeah.
Any other questions?
Yeah, I see over there that you define your diffusion coefficient as a function of mu A and mu S prime.
Uh-huh.
I also seen it defined in papers and textbooks solely based on mu S prime, based on the fact that in the diffuse regime, mu S prime is much larger than mu A.
Right, right.
So I don't know if there's any preference or ground truth or whatever.
Yeah, I don't know if there's ground truth. Vossen might actually have more to say about this, but yes, mu A, if much smaller than mu S prime, mu A is likely to have a small perturbation on that value of D. I don't know. Any thoughts on that? I don't have a good answer other than that.
Well, it's true. If you're purely concerned with modeling things in the diffuse regime, then the difference between adding or removing mu A is negligible. But I think the question is that if you want to push the use of the diffusion approximation to cases where you are saying mu S prime or mu A of 10,
Mhm.
then whether you include mu A or not does have an impact. And you get, I think, slightly better estimates if you include mu A. But you're right. Strictly, if you are in the diffuse regime, the inclusion or omission of mu A doesn't really make an impact.
Yeah.
Other questions?
So this is just such a convenient tool in your tool chest, right? You can make a device that's relatively simple and have a way of extracting mu A and mu S prime with some closed form analytical solutions under some specific conditions. And we'll go into some of those caveats in a moment. But I want to show you an example from literature of a group that's used the diffusion equation in this way. This is a paper from 2010, so it's a little bit old, but published in JBO. And it's from this group in the Netherlands, Sterrenborg, who's done a lot of work in SWIR, which I'm currently interested in. So what they want to do eventually is measure the optical properties and get water and lipid concentrations in tissues. And this is kind of their first paper that's exploring the model and the measurement technique. They followed up with many subsequent papers where they're trying to measure water and lipid concentration in breast margin specimens resected from a patient, try to determine where the tumor ends and begins. So they have this fairly close source detector separation, and they send in light from a lamp, broadband light from 900 to 1600 nanometers. They detect with a spectrometer, so they can detect diffuse reflectance at many wavelengths between that 900 and 1600 nanometer range. And here's an example figure. So data is shown in the little circles here, reflectance on the y-axis, so just really almost a raw measurement. And what they want is to take this measurement, in this case from subcutaneous fat, and extract optical properties in water and lipid concentration. And they use the same solution I showed you before. In the paper, it's shown just a little bit differently, but it's the same. So diffuse reflectance as a function of optical properties and source detector separation. And they're able to get mu A at all these wavelengths and mu S prime, shown on the same plot here. Notice that they're giving it to you in inverse centimeters. There's kind of a US-European divide. The Europeans are more likely to give you optical properties in inverse centimeters for some reason. But they have a lot of constraints and a lot of information. Note that this isn't spatially resolved, just one source detector separation, but they're measuring a lot of different wavelengths. So here they use Levenberg-Marquardt minimization. That's the wrapper, right? You solve the forward equation, but you're making these guesses. That minimization allows you to converge on optical properties. They really need some constraints here, though. If you took measurements at just one wavelength, let's say 1300 nanometers, there's a bunch of combinations of mu A and mu S prime that would give you that same diffuse reflectance. But if you take lots of measurements over lots of wavelengths, then you can start to say the absorption spectral shape has to follow a certain rule. In this case, they're saying absorption has to follow some concentration of water, they call it the fraction of water, times the absorption coefficient of water, plus some concentration of lipid times the absorption coefficient of lipid. And it has to look like that. It has to look like one of the combinations of water and lipid. They also said that scattering has to follow a power law spectral shape. So that's A lambda over lambda naught. Lambda naught is defined differently for different literature, can often be 800 nanometers, might be a different wavelength, and then the power minus B. And with those constraints, and absorption and scattering, you can converge on a more unique answer. You can also add constraints saying my absorption and scattering can only be so low or so high. My extraction of water and lipid have to be within a certain bounds if you know something about the tissue you're looking at. Yeah, Bernard.
Darren, in this case, do they take into account like the diameter of the fibers or the numerical aperture, or is that basically taken into account when they solve the fusion equation?
No, to the best of my knowledge, they didn't take that into account. And you might actually question their use of the diffusion approximation in this case. Remember, scattering is supposed to be a lot stronger than absorption. That's true in some cases, but look at this. They're on the same scale and very close source detector separation. So if I were to do this study again, I don't think I'd use a diffusion equation. And they have one line in the discussion about that. Yeah.
This is for the breast tissue, right? Or,
This isn't for breast. This is for, in this paper, they only measured ex vivo specimens, like porcine specimens.
Oh, okay.
But you're right in that they are preparing to do future measurements in the breast.
okay, well, my question kind of was, is this like accounting for the different layers or just like assuming it's homogeneous?
A major assumption here, homogeneity, that you don't have a layered structure or more complex geometry. You're assuming that everything is the same.
Oh, okay.
Yeah, major limitation. I'll talk a little bit later about multi-layer models, but tomography, which you could actually extract 3D renderings of more complex geometry under the surface, is a topic I won't have time to discuss too much today or tomorrow. But you're right to point out that limitation.
Other questions? This is a good discussion.
And
Bernard, I think you mentioned like the NA of like the detector and such. How would that affect these measurements? How would the detection geometry, like besides the source detector separation, affect?
I mean, I guess it would dictate a little bit of the collection of the light, right? If you had something that was completely forward-looking versus something that had an some angle that accepted, then it's going to accept more light if it's that angle is bigger, most likely. So then you get a higher reflectance or a lower reflectance depending on how much light that fiber is collecting, and also what's getting launched in as well, right? The light comes in straight versus it comes in at an angle. Angles are more superficial in general. So I'm not saying it has a massive effect. I'm just saying it could have an effect.
that effect will likely be amplified for very short source detector separation measurements, right? As you get the source detector further away from each other, some of that will be blurred out.
Yeah.
But, yeah.
Yeah, if I can amplify a little bit on that, is that, so we're within the context of the standard diffusion approximation. So as I mentioned in my earlier lecture, you can actually calculate the, within the diffusion approximation, the isotropic component plus the flux component, even across the boundary. So if you want to, you can actually figure out for a given numerical aperture, what is the partial angle that you would be collecting, and actually then calculate, just do a simple kind of partial solid angle integral, what fraction of the total backscattered first order fluence rate plus what fraction of the flux your actual fiber would be capturing. And you can get just and using that equation, it'll be a modification, but you can actually calculate those weights because you actually do get equations for the backscattered fluence and the backscattered flux. So you can actually, within the approximate framework of the diffusion approximation, you can still actually generate, maybe not an analytical, actually it'll be an analytic equation that actually does a little better than just looking at the total backscattered reflectance.
Nice. Thanks, Fasten. And then with Monte Carlo, you can, which we'll talk about a little bit more, you can dictate things like the NA and detector size pretty explicitly and very easily. Okay. Great questions. Keep coming. So diffusion equation is good for spatially resolved measurements when you have a lot of data. So broadband data, meaning lots of different wavelengths, many rows, many source detector separations, especially short and long, and spectral constraints. Otherwise, it can be difficult to disentangle mu A and mu S prime. Let's move on to the second, the modified Beer-Lambert law. Okay, I showed you this figure before. Remember, it's difficult to disentangle mu A from mu S prime, and it's difficult to know path length. So the modified Beer-Lambert law is a solution to the path length problem and the knowledge of the path length problem. So here's a typical processing pipeline. We compute what's called the differential path length factor, or DPF, based on initial assumptions about absorption and scattering of the tissue. Now, you could have a DPF estimate, I'll tell you more about what it means in a moment, based on diffusion theory, that's what you're seeing here, or you could have it based on Monte Carlo. But just practically, most commonly, DPF is based on an equation that's very similar to this, that's also based on diffusion theory, a function of optical properties, and you see rho in here, source detector separation. Remember our Beer-Lambert law? Light out equals light in times this exponential term. If we reorder this, bring I naught over to the left and take the natural log, get rid of this exponential term, you can just show it in this way. Okay, I'm showing you that because in the next line, we can solve for changes in mu A using what's called the differential modified Beer-Lambert law, introducing two things to you at once. First, the differential modified Beer-Lambert law and the DPF. But what I want you to see is, okay, look at this equation. Here's your Beer-Lambert law if you were just thinking about mu A. And here's the differential Beer-Lambert law, which is just telling you about changes in mu A rather than absolute mu A. And see L here? L is now being replaced by the DPF times the source detector separation. So this DPF is a modifier on your source detector separation. It's saying if I have this source detector separation, what's likely to be my average path length given optical properties between that source and detector, right? We know that rho is too short because photons will take a circuitous path. The DPF is your estimate of how long that path length is. So now it's just path length times a change in mu A. And we call the measurement, which is this natural log of light at one time versus an initial time, the optical density difference. Follow all that? Actually boils down to be quite useful and simple. You compute this DPF. The big thing is you have to come up with a DPF which requires you to assume some optical properties about the tissue. You're not measuring them explicitly. That allows you to get changes in absorption. Yeah, boss. No? Are you sure? Yeah. T-term in this case is time. And the reason it's time is because this is often used in a situation where, like in FNIRS, where you're measuring delta changes over relatively short periods of time, think seconds or minutes.
Yeah, so to clarify, time is not like you're putting a pulse of light in and you're looking at a time resolved signal, but rather looking at a system at time point one and looking at whatever change there may be in your chromophore concentrations at some later time.
Right, right.
Yeah, there in the expression for the change of the optical density, you are expressing the path length as you are like factorizing or separating the source detector distance and the DPF, right? So it looks like the DPF doesn't depend on the source detector distance, but then in the previous equation, you have it there. So
Yeah.
it's a bit more, okay.
It show up in both places.
Mhm.
And I guess the presumption also is that the DPF doesn't change with the change of mu A.
That's true. So this is there. I could go back. There's versions of DPF yet that don't incorporate the source detector separation because it's true. So what a lot of groups will do is say, okay, it's determined that for the head, the DPF is between something like 7 and 9. And it's not source detector dependent. So this expression I'm showing here does incorporate it, but yes, it's probably maybe not the most commonly used one.
Well, but not only not source detector dependent, but it doesn't change with the perturbation in the tissue
Right.
system, which also kind of makes sense in the context of a standard diffusion approximation.
Yep.
because
Yep.
Scattering is what's dominating.
That's right.
Other questions? These are really good questions.
And then if you can get delta mu A at two wavelengths, 750 and 850 are common, you can use Beer's law to get back to delta oxy and deoxyhemoglobin. So this is also assuming that mu S prime is not changing. It's also assuming that mu A doesn't change dramatically. right? It's for relatively small perturbations, but used all the time for FNIRS, where we have these optodes on the head or a collection of optodes, and we look at changes in oxy and deoxyhemoglobin that tell us about functional activation. And folks looking in this area are typically less interested in knowing the absolute optical properties and more interested in knowing where the functional activation is occurring. So take a look at a figure from this paper from Nature Photonics from a few years ago. Here they're looking at what's called high density diffuse optical tomography, but it's a version of FNIRS. And they are showing when a volunteer is hearing words, this part of the brain is lighting up, versus when they're reading to themselves, this part of the brain is lighting up. Right, so here we can say the DPF is approximately X, like 5 to 7, and it's enough to know where this is occurring. Sometimes you want to know the magnitude, but you don't need to go all the way to mu A and mu S prime. Okay, so modified Beer-Lambert law, good when you just want delta chromophores, requires assumptions about mu A and mu S prime. That's equivalent to saying requires an assumption about the DPF and assumes only mu A changes, at least in the forms I showed you. Okay, but when doesn't diffusion theory work? Back to your question. So when you're very close to the source, so here's rho 0, here's diffuse reflectance. You can see if you plot what diffusion theory tells you versus Monte Carlo, there's divergence, and the divergence is the worst close to the source. So often you don't want to use this for close source detector separations. Yes?
For that figure, do you have a sense of what optical properties they're considering?
I don't have those available to me now. It's from Irving and Sergio's textbook, but they're choosing a set of optical properties. And I'll show a figure of yours, Vasan, in just a moment, where we do know the optical properties. But just visually for this tissue, you can see that divergence gets worse closest to the source. This is that mu S prime over mu A for the 3 different tissues. That ratio here is 10, and you can see, especially in the shortwave infrared, but also in some parts of the visible for some tissues, mu S prime over mu A is not large, not greater than 10. Remember, our diffusion approximation is valid when mu S prime is much larger than mu A, or mu S is much larger than mu A. So here's one of your figures, Vasan. So in this figure, source detector separation versus diffuse reflectance. Monte Carlo is shown in dots. Standard diffusion approximations shown in solid line. And then we're parsing this out with different mu S prime over mu A ratios. So you can see when this ratio is high, 110, red and green, they don't diverge from each other as badly, but there's still divergence close to the source. But as that value gets lower, like in properties you would expect to see in the shortwave infrared, there's much more divergence. We showed this in my group in a different way for a specific tissue, for brain tissue, for example. You're looking at mu S prime over mu A. So wherever you see the blue shading is where that value drops below 10. And the red line tells you the divergence or the error in diffuse reflectance using the diffusion approximation versus Monte Carlo. That error really, really increases as you go out into the shortwave infrared. So the diffusion equation and modified Beer-Lambert law are typically not good solutions for the shortwave infrared, depending on the tissue. So then you might want to use a Monte Carlo-based inverse model. And you had a whole lecture on Monte Carlo, so I won't explain what it is, but I do want to show you some examples. This is a paper from Nimi Ramanujan. So she's at Duke now. It's a well-cited paper, 469 citations as of yesterday. And it's a 2-part paper. In the first one, they're really talking about their forward and inverse model. And the goal here, again, is related to breast cancer. The second part of the paper is them extracting optical properties, chromophore concentration, using mostly visible wavelengths of light to try to diagnose different breast tissues as either cancerous or non-cancerous. In their forward model, they start with absorber concentrations. Here they're thinking about hemoglobin and beta carotene because they're at shorter wavelengths. Beta carotene shows up. Scatter size and scatter density. They use Beer's law to get at mu A as a function of wavelength. See that natural log of 10 that shows up again, that 2.303 times the extinction coefficient times the chromophore concentration. You get scattering and G values from me theory. You have a chance to talk about me theory in the short course? Okay, good. And then Monte Carlo will take those values and convert them into diffuse reflectance. And then again, just like with diffusion approximation, they wrap this up into an inverse solver where you have measured diffuse reflectance from your instrument compared to the modeled diffuse reflectance for some given input parameters. You look at the difference. Here they're using Gauss-Newton nonlinear least squares optimization to converge onto the optical properties of the tissue. And you might ask, doesn't this take forever? Monte Carlo simulations are slow. And in this case, they're using what's called scaled Monte Carlo. So you can run a smaller number of simulations and scale those results to get results, diffuse reflectance at many different optical properties, even ones you didn't simulate. And Vasan is an expert in conducting simulations like that. But this is an early example of that. And so let's see what they did with it. Here's part 2 of the paper, a few figures. Here's a measurement, diffuse reflectance versus wavelength from 400 to 600. They took these measurements from the spectrometer, converted them into optical properties, and now you're seeing mu A for malignant tissue in the black solid line and adipose tissue, non-malignant tissue. You can see the absorption are different. The scattering is different. That's shown here, reduced scattering coefficient. And then they take these absorption coefficients and separate it out by malignant, fibrous, adipose tissue. Here you're looking at beta carotene and total hemoglobin. You can see, for example, the hemoglobin saturation is different for malignant tissue from fibrous from adipose tissue, right? So again, those are the biomarkers that they're using to help diagnose whether there's cancer or not in the measurement volume. Yes?
Is it all like based on Monte Carlo? They don't have, I'm sorry. This is all based on Monte Carlo. They haven't done any like samples of the breast, of the malignant breast tissue.
Oh, this is actual measurements.
Okay, this isn't the Monte Carlo.
Well, they're processing the data using a Monte Carlo-based inverse model. So they actually take the spectroscopy measurements and then on the processing side in MATLAB, they're converting into optical properties and then chromophore concentrations using that Monte Carlo-based inverse model.
Okay, I'm just curious because we're doing a PBL proposal and I'm trying to do that for something in my lab and I don't know how to do it.
Yeah.
So I'm based on the stuff you've already presented, I feel like the first 2 options wouldn't really work for
Okay.
my application, but this might.
Yeah, yeah. And so who could tell me why they're using Monte Carlo in this case, as opposed to the diffusion theory? Well, this still is assuming homogeneity. You could develop a more complex inverse model that's multi-layered, but in this case, it's assuming homogeneity.
Does it just fit better than diffusion fitting, I guess?
It probably does fit better, but why? What about this might lead you to... You can see them right here, right? So these are in the same units.
Yeah, the
Yeah.
relation between the absorption and the reduced scattering coefficient.
What about it?
They are too close to each other and the diffusion approximation doesn't work quite
Right.
well.
In this figure, you can see the extracted optical properties, right? Especially here at really short wavelengths, absorption is up above 10 inverse centimeters. It's about the same scale as scattering, right? So your mu s prime over mu a is in some cases close to 1, and that's kind of no-no territory for the diffusion theory. And that's because you're in the visible, right? Not in the near infrared anymore. Visible absorption is really, really high.
So for the hemoglobin saturation plot, is that the oxygen saturation? Are they talking about that?
I believe it's the oxygen saturation. So they're extracting oxyhemoglobin concentration and deoxyhemoglobin concentration, then oxy over oxy plus deoxy. So
Yeah.
tissue saturation, yes.
The numbers are over 100.
Yeah, some of them are for the error bars. Well, you actually don't know from the error bars, right? Because we don't know which way. You see the mean's not over 100, but the error bars or the values might be such that the error bars are are peaking over 100, even if you don't have individual measurements. But yeah.
Darren, do you remember in this paper what the source detector geometry was? I guess they're just doing some sort of diffuse reflectance at one source detector
Yes.
separation.
Yes.
Is this a fiber-based measurement?
I believe it's a fiber-based
Yeah.
measurement. Yes.
So I just want to give a bit of an editorial. I think some people would actually take a little offense for saying this is a Monte Carlo based inverse model, only because in a Monte Carlo simulation, you actually, to get your estimate of your reflectance for a given detector, you collect the photons and you derive a mean and variance. And they do that, of course, for the baseline, but then they scale. And
Yeah.
in the scaling, what you do is that you take whatever detected set of photons and you assume that all of them had the mean value and you scale based on the mean value. And so that, and depending on the magnitude of the scaling, how far you have to scale away from the baseline, your variance can really explode and actually your mean can change. In a rigorous Monte Carlo, you would examine the entire population of collected photons and scale each of the photons, detected photons separately. And I don't believe they did that in this paper.
I don't think so either.
So I just suggest, this is more just to educate. I'm not criticizing the paper. Paper has great results, but just be aware that when you scale based on a mean of a population of collected photons, your scaled estimates are not rigorous from a Monte Carlo perspective. If you want to do it rigorous, you should scale the weight of each and every photon that was collected in that baseline Monte Carlo.
Fair enough. And Vasan's the right person to know that nuance, and also the right person to take offense to something like calling it a Monte Carlo. Like, you really have to care about the methods in order to be offended by that, but appropriately so. Yes. Yeah.
Do you mind go one slide back?
Sure.
I'm just wondering, like in this inverse model, will they also have to add constraints to really remove like other solution that can give you the same results, like local minimum, basically?
So I actually don't remember what other constraints they added, if any, including power law or any constraints related to the chromophores. But remember, they do have a lot of spectral data. So we'd have to go back and dive in. I'm not sure offhand.
Okay, basically you're saying like you have like multiple spectral data to fit the same solution that can like act as a constraint.
Yeah, you're fitting all those measurements simultaneously.
Right. Yeah, that makes sense. Thanks.
It's a good question, though.
Darren, just heads up, I think we can certainly go forward with the rest of the inverse models, but I think we should get to SFDI at some point.
Yeah.
And
Well,
we have, I think, about 20 minutes.
it's been a robust discussion.
Yeah, so
Yeah.
maybe we want to accelerate to maybe even jump, because I think you've given them enough nuance to approaches for the inverse problem, and maybe we want to introduce the other domain.
Okay. Or would you guys like to skip lunch and just talk about diffuse optics
Well, we got our
instead?
academic career panel.
Oh, that's right. Yes.
Right.
Okay. Well, one of the things you need to know if you're in academia is that you should never stay on time for a talk, right? You need to really practice that to get that right. Okay, let's see if I love the discussion, though. I almost don't want to rush it just because we want to saturate in all this, so to speak.
But they do have some SFDI in the afternoon lab.
Yes,
And so we
okay.
need to kind of give at least a baseline of knowledge so that they can
Got
pursue
it.
that intelligently.
Got it. So just finishing up on the spatially resolved, other ways you can tackle this, right? You can build lookup tables. This is from a paper from James Tunnel's group. And is somebody from James's lab here? Yes, yeah. So their paper from 2013, you can see this is really nice visualization. Mu S prime versus mu A versus diffuse reflectance. You can immediately see that for a given diffuse reflectance measurement, there's a lot of different mu A, mu S prime combinations that will give you that same answer. But if you take measurements at multiple source detector separations or across wavelengths with the constraints, you can get to a more unique answer. Okay, so Monte Carlo-based inverse model is good for when the diffusion approximation doesn't work. Small rho, small source detector separation, SWIR invisible. And then the last one is related to neural networks. And it's really dependent on the other. You can train a neural network based on simulations from fusion approximation or Monte Carlo, but a deep neural network, for example, gives you a very compact, very fast solution that you could put, say, in an embedded system like a wearable. And I was going to show you an example from my lab for this little SWIR wearable probe. If you're interested in learning more about it, I'll talk a bit more about it tomorrow, but I'm going to skip through it to get to the spatial frequency domain imaging. Okay, so for spatially resolved measurements, we had our source and detector separation. What we have, this relationship between distance and our measurement, which we call the spatial point spread function in the spatial resolved domain. The shape of this curve is dependent on mu a and mu s prime. One can take a Fourier transform of this point spread function and get to the next domain I'm going to talk about, the spatial frequency domain, where we switch our axes. Now the x-axis is spatial frequency in k, defined as inverse distance, inverse millimeters in this case. So inverse distance meaning number of cycles per unit distance. And we have a pattern of light that we project onto tissue. So these are cousins related through a Fourier transform. This is the newest of all the diffuse optical techniques, but it's most related to the spatially resolved measurement, which is why I covered in this lecture. And here's how it works. We project spatial patterns of light onto tissue, non-contact, measure the remitted light with the camera. You can see this is wide field. This is the back of someone's hand. You see these different spatial patterns of light at different spatial frequencies, which is shown in the bottom. So DC is just planar illumination, higher and higher spatial frequencies. We typically measure at least one low spatial frequency, typically 0 DC, 0 inverse millimeters. That's same as saying no pattern. And then at least one higher spatial frequency. We also project that pattern at different phase offsets. We shift it in order to use this demodulation algorithm. What we want to know is how the tissue is modifying the modulation of that pattern. Is it diminishing its amplitude? You could theoretically measure how it shifts the phase of that pattern, but that's a difficult actual measurement to take. So usually we just measure the amplitude change. And to get that, you want the envelope of that pattern. You can use this demodulation equation. I'll show you more about that on the next slide. But once you demodulate and calibrate, you can get at diffuse reflectance as a function of spatial frequency. So here's the same back of the hand for DC and higher spatial frequencies, shifting through time here as we go to higher and higher. We can take pixel by pixel the diffuse reflectance at a low and high spatial frequency, or many different spatial frequencies, use an inverse model and calculate optical properties at each pixel in the image, and get maps of mu a and mu s prime. See the superficial blood vessels that have high absorption, scatterings telling us about the microarchitecture of the tissue. And of course, if we do this at multiple wavelengths, we can use Beer's law and extract chromophore concentrations and use those as our biomarkers. Let's dive in a little bit more. Here's our measurement again, a projected pattern, different spatial frequencies, different wavelengths of light. We project for a given spatial frequency at these three phases. If we take a line profile through those phases, you can see them shown here. 0, 2 pi over 3, and 4 pi over 3. What we want is the envelope of these measurements. There's an analogy here to AM radio that sometimes students don't get because we don't use AM radio anymore. But back in the day, you tune the dial to a given carrier frequency, right? That's what the station was, and typically in the megahertz. The carrier wave gets modified by voice information or singing information. Let's say your carrier wave is at 100 megahertz. You're modifying in small perturbations in the kilohertz on that megahertz wave. The analogy here is the spatial frequency is the carrier wave that transmits the data, and the tissue is modifying that carrier wave, changing its modulation. And it's doing that pixel by pixel. And you can use this, the demodulation algorithm, just shown here, to get that envelope. the information imprinted onto the carrier wave by the tissue. And it's that information that encodes absorption and scattering. So, showed you this before. We had the spatial modulation transfer function. We measure at a low and a high. We get diffuse reflectance. We can kind of get the shape of this modulation transfer function, which is going to be dependent on the optical properties of the tissue. How do you solve the inverse problem? There's at least 3 different ways. One is the first I showed you, the diffusion equation, same thing. When I was first at BLI, SFDI was fairly new. I worked on it a little bit, but people in my office with me were solving the diffusion equation pixel by pixel for SFDI images. And it took hours and hours, days, right? Because this inverse solver is kind of slow because it's iterative. A faster way is to pre-compute a lookup table using either the diffusion equation or Monte Carlo, where you build up the relationship between diffuse reflectance measured at a low and a higher spatial frequency. You can visualize this lookup table here. The ISO lines correspond to values of absorption and scattering. Like this line right here, this dashed line is mu A equals 0.003. And then the more vertical lines correspond to given US prime values. You can also train a neural network based on these results to get a much faster answer. We showed that a few years ago in an optics letter paper, where we compared, say you had an image that's 100 by 100 or 696 by 520, the iterative method could take you all day. This is in seconds, like 40,000 seconds. The nearest search, which is the lookup table-based method, much, much faster. And the deep neural network is faster still, less than a second. And so just to link these two together, here's our spatial modulation transfer function. We're taking measurements at 2 different spatial frequencies. Where those measurements, the measurements are on the X and Y axes, where they converge, should hopefully give you a unique mu A and mu S prime. And you have more information, so to speak, than in the spatially resolved domain, if you choose your spatial frequencies appropriately. This is from one of the original papers by David Cucci and Bruce Stromberg, kind of explaining how SFDI works. It's a really nice demonstration of how spatial frequency measurements allow you to extract optical properties. So here's the experiment. You have a liquid phantom. You keep mu S prime constant and you titrate mu A. Each one of these lines is for a different absorption value. Scattering is all the same. Spatial frequency on the x-axis. You see at low spatial frequency measurements, changes in absorption really affect your measurement, your diffuse reflectance measurement. At really high spatial frequencies, changes in absorption almost don't affect your measurement at all. Let's do the opposite experiment. Let's keep absorption the same and titrate scattering. Each of these lines is associated with a different scattering value. See, both at low and high spatial frequencies on average, you're going to affect the diffuse reflectance. This gives you enough information typically to extract absorption and reduce scattering. Why does mu A seem to have such a small effect on diffuse reflectance at higher spatial frequencies in this part of the curve? There's different ways to think about this. In fact, Vasan and I were talking about 2 different ways we think about this before the lecture. But it's not a trick question. You can think about an analogy back to the spatially resolved domain, if that's helpful. Any thoughts?
I just have a guess. Maybe like all the photons that would be absorbed have been absorbed by that point. So then all the photons left are just like left to scatter.
Okay. Well, let's riff on that. Like, why didn't it, remember at very short source detector separations, why didn't an absorption change our measurement? Why was it only scattering? What was the reason for that example? Yeah.
So absorption is so much smaller than scattering.
Which
So-
means that for a given path length that a photon travels, right? You're
Yeah.
going to have different probabilities of absorption versus scattering.
Right.
Can we link that back to why at a high spatial frequency, then might you not be sensitive to changes in absorption? Mhm. It's kind of similar, right? High spatial frequency
Can
is,
you repeat your comment? Which is a good one.
Sorry, when it's closer, when it's at a higher frequency, the light bands are closer together. So it's almost like you're having two like the source and the detector very close together.
yeah, that is one way I typically think about it. I also think about it, you must be segmenting the photons, right? At higher spatial frequencies, you must be sensitive to photons that have traveled shorter path lengths. Whereas at low spatial frequencies, you're sensitive to photons that have traveled longer and shorter path lengths. Boston, do you have another way you like to think about it?
Well, if people get that way, I don't want to confuse them.
Well,
Are
people understand in different
satisfied
ways.
by that answer? And so the way I think about it is that, remember, Darren talked about a carrier wave. And basically what you're trying to figure out is, of the reflected photons, which of those photons still maintain that frequency? And so if you have two bands that are really far apart, it's going to take a lot of interaction of those photons with the tissue over longer path lengths in order to blur those two lines so that that frequency no longer exists. And that means you have to have a lot of scattering events, which means you have to have a lot of path length. And that's where you then become sensitive to absorption. But if the lines are really close together, they don't have to interact with the tissue that much before they blur because they're so close together. And so the photons that still retain that carrier frequency must have not traveled that much in the tissue. Because if they traveled more in the tissue, they'd get blurred and the information is lost. And so the high spatial frequency is uniquely more sensitive to scattering because you're not going to have any, well, I wouldn't say more sensitive, meaning that it's more specific to scattering because there's no way you're going to have sensitivity to absorption because the information is lost over much, or that carrier frequency is lost because of spatial dispersion, scattering-based dispersion.
Yeah. One kind of special thing about that demodulation algorithm is, you think you're illuminating all these photons. Your measurement has to incorporate all those photons. But actually what the demodulation does is throws out a lot of information from a good portion of the photons. So out here at high spatial frequencies, you're only sensitive to or really listening to, whatever analogy you want to make, to some small portion of the detected photons. And in this case, it's photons that have traveled shorter path lengths, right? And that's a little bit conceptually more challenging to think through than just having the source very close to the detector, where you can see a bunch of the photons are just going somewhere else and not to your detector.
Another way to think about it is that the photons don't have to penetrate that deeply before they get kicked out of the AC and go to the DC background.
Yeah.
Because once you lose that contrast between adjacent lines, it's basically a DC background over which you have a small carrier wave that's riding on top. Whereas on the low spatial frequencies, you still have more comparable magnitudes between an AC amplitude over a DC background. I think that's another way, maybe.
Yeah, yeah.
Hector.
Yeah, so according to what we have been discussed so far, it looks like you can set the sensitivity to depth with the spatial frequency.
You got it, my friend. So what would you guess as you went from low to high spatial frequencies about imaging depth? I'll call it imaging depth rather than depth penetration. So at a low spatial frequency or high spatial frequency, would you be more surface enhanced is one of the ways we say it, more superficial? Mm-hmm.
I mean, I think it's just like the photons are leaving at a like a smaller short source detector separation, and typically penetration is correlated to the source detector separation.
Yeah.
value idea. I know obviously
It's an analogy,
yeah.
but yes. Yep.
Yeah. That was my-
And in fact, if you keep taking this into the kind of out of the diffusive regime, there's just a technique called structured illumination microscopy, right? Where you can have pattern illumination, do the same demodulation. It gives you a higher spatial resolution microscopy image because you are getting rid of more and more of the multiply scattered photons, getting a high resolution image. It also comes to a fundamental issue that we think about in SFTI. You really need a low and a high spatial frequency in order to extract optical properties, but that low and that high spatial frequency are sensitive to different depths in the tissue, right? So that's a partial volume effect that in many circumstances we hope doesn't cause too large of an error. Okay, that was great. Now, if you pick the wrong spatial frequencies, your lookup table, this is just the relationship between a low and a high spatial frequency for a given tissue, cannot work so well. So here's DC plus 0.025. So very low and a kind of low spatial frequency. You see the relationship between those measurements is almost linear. It's going to be hard to separate absorption from scattering. You want to be in a situation like this where those isolines are a bit more orthogonal. And if you go too high, DC plus 0.5, this becomes challenging again too, because you're compressed all the way to one side of the dynamic range here. Diffuse reflectance just falls off with spatial frequency. Really difficult practical measurement to take, because for a given change in optical properties, it's going to perturb our D very little. And you guys know what a cuff occlusion is? It's one of kind of the experiments, in vivo experiments we do over and over again when we test a device, put a blood pressure cuff, block, sometimes venous and arterial circulation. This is what an occlusion experiment looks like if you extract oxy and deoxyhemoglobin. Oxy goes down during the occlusion, deoxy goes up, and then it flips when you release. But this is an example a postdoc of mine came up with a few years ago, where if you choose with SFDI and image the hand during the cuff occlusion, you get this beautiful result if you use 0 and 0.1. But if you use 0.05 and 0.1, two spatial frequencies that are too close to each other, the uncertainty in the optical properties of the chromophore extractions balloon up, and it's more difficult to detect that change in physiology here in that state. Yes.
I just wanted to ask, is there like, because I'm guessing how you have to space them depends on your optical properties.
Yes.
Is there like a calculated way to do this, or is it kind of like a guess and check that you do? How would you pick your frequencies?
So I could answer that in a number of ways. I will say that the SFDI community typically is landed on something like DC and 0.15, sometimes DC and 0.1, sometimes DC and 0.2 for a lot of different tissues. We published a whole paper a few years ago about spatial frequency selection and gave results for different tissues with different optical properties and what the uncertainties would be in the optical property extractions based on different combination of spatial frequencies. So you can take a look at that if you're interested in it, but that DC and 0.1 gets you in the ballpark for many tissues, yes. Yeah. When doesn't diffusion theory work for spatial frequency domain? Remember, we could solve, or somebody could, I couldn't, solve the diffusion equation for the spatially resolved domain. Well, you can do that for the spatial frequency domain as well. It turns out at high spatial frequencies is where you get that divergence from Monte Carlo. And the high spatial frequencies are sort of like very close source detector separations. Well, it makes sense that they would diverge out there. So SFDI, some advantages are that it's simple-ish. You need to project these spatial patterns somehow. Non-contact wide field, quantitative mu-A, mu-S prime in a mapping format. Some potential disadvantages are relatively shallow. You're going to use this for skin or the superficial part of a resected specimen. And the analysis is more complex typically than spatially resolved measurements. Not terribly complex, but just more complex. We have an open SFDI project. I know I had to finish up here. We're right at the end. Can I go a few more minutes or no? Yeah, okay. An open SFDI project, opensfdi.org, What it has on it is basically plans, parts list, how to build your own SFDI system, even some example pseudocode and to do processing, example processing code. This is a collaboration between my group and several other groups.
You'll see a system that leverage the open SFDI.
Nice.
In the first HD.
Okay, excellent. We also published a companion paper in JBO where we compared the OpenSFDI system to Modulim's system. Modulim is a startup company that came out of Beckman, makes a commercial SFDI system. And so we use that as sometimes our gold standard device, these OpenSFDI systems. And my lab at the University of Maine and a few others were compared to that system at these 3 locations. You can see that multi-system validation here. And then I was going to tell you about how we've extended SFDI into the shortwave infrared, but I think I'll skip that and maybe save it for tomorrow so that we can go ahead and eat lunch. And I didn't even get close to telling you about wavelength selection. I thought maybe I'd have to sacrifice that, but I'm going to go ahead and sacrifice that. Okay, our learning objectives at the beginning were to gain an intuitive and quantitative understanding of how diffuse optical techniques work in practice. Hopefully we've gotten close to that for these first 2 versions of the diffuse optical techniques. Tomorrow we'll talk more about the other 2 techniques and their advantages and disadvantages. We covered these topics in some detail. Now tomorrow we're going to start with some bookkeeping. I'm going to tell you more about chromophores, real-world resources, literature sources where you can get extinction values for chromophores. We're going to talk about time domain and frequency domain. We're going to compare all 4 techniques, talk about when to use one versus another, and go through some example applications. All right. Thanks, everybody.
Great lecture, Darren. You covered a lot of ground, but I think in a very digestible manner. So thank you for that. A lot of topics. Any questions? Oh.
So I think at some point you mentioned that you would talk about layer media.
Yeah.
So I don't know if you will talk about this tomorrow or.
Yeah, it's actually one of the things I jettisoned a little bit when I skipped through a bunch of slides here.
Yeah.
But for inverse models, there are analytical solutions for multi-layered media. 2-layer is quite doable. 3-layer is doable, but more difficult. Monte Carlo, you can do the same. You can imagine, though, if you have separate optical properties for these different layers and thicknesses, solving for all that at once increases your requirement to have more and more measurements that allow you to get to a unique solution. We've certainly had applications where we're imaging in the skin. We define what the skin optical properties are from literature to try to extract the optical properties of deeper tissue. That's one way to do it. And then, of course, there's a whole world of tomography, which you can do with spatially resolved measurements, FNIRS, even SFDI, where you take enough measurements to reconstruct a 3D model of optical properties. So for example, you could have a 3D model of a tumor that's embedded. Yeah, yeah. People haven't developed any new ones recently, but maybe about a decade ago, Soren Konecki, who was a postdoc here, did a lot of work in tomography with SFDI.
Other questions for Darren?
I'm trying to find the slide number on here, but it was the one where we were talking about why we used Monte Carlo instead of diffusion when we're looking at the unhealthy versus healthy tissue. And
Yeah, or is it the other one? This one? Or
no, it was
further back?
Yeah, keep going. It was like the let me find. Yes, that one. It's I don't know what slide that is.
It's the one after the one you showed.
After NIMIs. There's
After
NIMIs.
this one. The one before. Yeah. I was a little confused on why we didn't just use diffusion, because I know you can use diffusion in the visible in the visible wavelength field usually. Is it because the reduced scattering is at 9 and the absorption coefficient is at 10? Is that why? Or is it because the differences between malignant and adipose?
So take just one of these tissues, for example, like let's take malignant. And so you can see mu A here and mu S prime here. And just compute in your head an estimate of what mu S prime over mu A would be at these different wavelengths. Right? What you'll see here is, around 425 or so, absorption is actually higher than scattering. Right? So you're way out of the diffusive regime in this case. The Soré bands of hemoglobin are so strongly absorbing there that you're sort of like the SWIR in that absorption can be on the same scale or even stronger than scattering. And one of the assumptions for the diffusion approximation is that scattering has to be quite a bit higher than absorption.
Okay, so that's like dependent on the tissue that we're looking at.
Absolutely, it's
Yeah.
dependent on the tissue, the makeup of the tissue, right? If you had a tissue with much less hemoglobin, maybe it wouldn't be as bad.
Mhm. Is scattering usually higher in the visible? It's usually higher in the visible, so then diffusion just wouldn't work for a lot of cases in that way. Yeah, because we want scattering to be higher than the
You
absorption.
want scattering to be
Yeah.
higher. Problem with the visible is absorption is so high too,
Yeah.
right? So
Okay.
diffusion approximation works maybe the best in the near infrared, is the
Oh,
way I'd
okay.
say it.
Okay.
And works less good, shorter and longer.
All right.
Speaking in generalities here, like skin is an exception. It doesn't have as much hemoglobin in it, typically just the skin layer. So fusion approximation can work at longer and sometimes shorter wavelengths.
Is it like that's like is it because the melanin content is really high absorbing earlier on?
Melanin does absorb stronger at shorter wavelengths. It sort of looks like the wavelength dependence of scattering in that it increases absorption at shorter wavelengths.
Okay.
But just skin, all the way at the surface of the skin, you just have capillary loops. You don't have a ton of hemoglobin, which is that's what's causing this super strong absorption here is hemoglobin and beta carotene to some degree.
Okay. Thank you.
These are resected specimens, so they may not have melanin on them at all, right?
Other questions?
So in the case where you have close source to detector distance, like which method would you think is best? Would it be the Monte Carlo or would it be something else?
Yeah, Monte Carlo. I mean, that's really your diffusion theory and Monte Carlo. And the other things like the lookup table and the deep neural network, they're still based on either diffusion or Monte Carlo. That's how you train those or build those tables.
Okay, thank you very much.
Darren, I want to ask a question going to Dick Sternberg's
Yeah.
work. If you can pull up that slide. I noticed that in that paper, they recovered mu sub A from, I guess, a spectral consideration of the reflectance. I think traditionally, or what has been done in the past is that groups recover mu S prime and mu A at each wavelength. And, you showed the matrix equation of mu A, how you can get mu A at a given wavelength, that's the linear sum. So I just wanted to get your thoughts about those two ways of doing things. First, you can have a processing stream where you first recover mu A and mu S prime at each wavelength, and then you do the, say, linear regression or least squares matrix assessment of what the concentrations of each of the chromophores are. Or it seems like here they looked at everything at once across the whole wavelength domain. And can you talk about maybe the pros and cons of those approaches and, which approach do you adopt in your lab?
So we do some CW, but I think the second approach is the way to go in this case. I don't think you have enough information at a single source detector separation to extract optical properties in chromophores at each wavelength independently, right? Depending on the noise in the system, you are not going to have continuous spectra.
For a single, but say you had multiple source
Yeah.
detector separation, or you had multiple spatial frequencies.
Right.
And you can do either way. You can either recover mu A and mu S prime at each wavelength and then get chromophores, or do you want to do the inverse problem directly across all wavelengths?
Yeah, good question. I feel like that is so context dependent on the tissue and the optical properties you're measuring. What we'd probably do is run some simulations to kind of get our head around which one of those works better. We've done it both ways. And for CW and spatially resolved, we tend to fit everything all at once. But for SFDI, we'll often calculate the optical properties independently and then do chromophores.
Any final questions?
So for SFDI, we kind of treat each pixel independently from the next one. What are your thoughts on that? When you have like, let's say a really absorbing one versus like non-absorbing one right next to each other.
Yeah. So this is a topic I think that's come up as long as SFDI has been around, right? It's a interesting one to think about. Those pixels are not independent from each other, right? If you think about projected area of the pixel of the camera onto the tissue, likely your photon path length is going to cover the equivalent of multiple pixels. So they're not truly independent, but you're solving the inverse model independently at each pixel. What can one say? Maybe there's a lot to say. I sort of come back to the fact that we can see heterogeneities, but they're blurred, right? Because you're in the diffuse optical technique. You see not sharp transitions, but they're blurred because you're diffusive. Higher spatial frequency should make you less blurred, less interaction of pixels by pixels. But I don't know, that's my empirical way of thinking about it. I can tell that Vasan has something to say about it, I think. No? Nothing to say about
I
it?
think we should thank Professor Roblyer for, that's what I have to say. I think we should thank Professor Roblyer for an incredible lecture. Thank you.
Okay.
So we have our academic career